I hope you're doing well. Today we're gonna do 9700 paper 1-1 Major 2025. So question 1, which statements about light microscopes are correct? To calculate the magnification of a light microscope, the eyepiece lens and the objective lens magnifications are added together. This is wrong. To find the magnification, use the formula magnification is equal to image size upon actual size. The resolution of a light microscope is limited by the wavelength of light. This is correct. And the divisions on the scale on a stage micrometer are closer together than the divisions on scale on an eyepiece graticule. This is also wrong. We can figure it out from this image over here that the eyepiece graticule are much more closer together than the stage micrometer, and we use the eyepiece graticule to calibrate the stage micrometer. Hence, our answer for 1 should be C. Question 2. A student measured the width of a mitochondrion, electron micrograph an animal cell, magnification is given, the width is given, we need to find the actual width. So since all of these options are in micrometers, we have to multiply this 6 into 1000 to convert it into micrometers and divided by 9600, the magnification, in order to get the actual width of the mitochondria, which is 0.625 micrometers, and the closest answer we're getting is B, 0.6 micrometers. Number 3, the photomicrograph shows a section of the stem of a plant. Which statement could describe Q? So Q shows the xylem over here. So the cell wall contains pits. That is correct. Xylem does contain pits. It contains cellulose and lignin as their dead cells, and is connected to adjacent cells by plasmodesmata. This is wrong, because plasmodesmata is a characteristic of living cells, and you need cytoplasm for there to be plasmodesmata. Hence, this is wrong, and our answer for 3 is A. 1 and 2 only. Moving on, we got question 4. The diagram is taken from an electron micrograph of a cell that secretes enzymes. Where are the polypeptides for these enzymes made? So the polypeptides are made in the rough endoplasmic reticulum, which is C. Which comparisons between a typical bacterial cell and a typical plant cell are correct? Cytoplasmic DNA, yep, this is correct. Nuclear DNA, this is also correct as a comparison between those two. No smooth ER is present. Yes, they don't have any membrane bounded organelles. Smooth ER is present, that is correct. In plant cell, peptidoglycan cell walls, correct. Cellulose cell walls, correct. 80S ribosomes, 70S ribosomes, this is wrong. Bacterial cell has 70S ribosomes, while the plant cell has 80S ribosomes. And of course, the mitochondria and the chloroplast in the plant cells have 70S ribosomes. So, but of course, bacterial cell does not has 80S, it has 70S ribosomes, so this one is wrong. Hence, our answer for 5 should be A. And by the way, cytoplasmic DNA means that it is located in the cytoplasm of a bacterial cell, which is correct. Moving on, question number 6. Diagram is showing a virus. So of course we know that this is the DNA or the RNA, which is surrounded by a protein capsid layer, which is made up of capsomeres, the monomers, which makes up capsid, and this is made up of protein. So Y should be our proteins, and W could be either DNA or RNA. We'll look into it. X is, of course, surrounding the protein capsid is always a lipid envelope. Hence, X should be our phospholipids or lipid envelope, and W gives us DNA, and Z are the protein spikes, which are glycoproteins majority. But since the major part of glycoproteins is made up of proteins, hence we will consider that mainly as proteins, because it doesn't have to be all those glycoproteins, but even if it is glycoproteins, it's mainly made of proteins. Hence our answer for 6 should be B. Number 7, the iodine test and Benedict's test were carried out on a sample of starch solution. Amylase was added to another sample of the starch solution and incubated for 10 minutes. The Benedict's test was then carried out on the sample. What were the results of the test? Iodine test before adding the amylase. So before adding the amylase, the starch hasn't been broken down, so the iodine test will be positive. Benedict's test before adding amylase. Benedict's test will give a negative response, as it's still starch there. There's no reducing sugar present. Benedict's test is only for reducing sugars. Benedict's test after adding amylase, since starch will be broken down into maltose, so reducing sugar's presence, so it will give a positive test. So our answer for 7 should be C. Which row about alpha glucose and beta glucose molecules is correct? Carbon atom on which the OH position is different, so that carbon atom is 1, as we can also see from this screenshot over here. This is carbon number 1. In alpha glucose the OH is below, however for beta glucose the OH is above. And cellulose contains both molecules. That is not right. Cellulose only contains beta glucose and not alpha glucose. Hence our answer for 8 should be A. 9. Which molecules are monosaccharides? Ribose, of course, monosaccharides. Glucose, also monosaccharides. Deoxyribose, of course, monosaccharides. Sucrose are not monosaccharides, they're disaccharides, as they're made up of glucose and fructose. Hence our answer for 9 should be A. 1, 2, and 3 only. 10. Many animals use triglycerides for long-term energy storage. Which statements are correct? Additional functions of triglycerides: they provide buoyancy in submarine animals. That is correct. They are the main components of cell membranes. That is wrong, because triglycerides only contain 1 glycerol and 3 fatty acids. However, the main components of cell membranes are phospholipids, which contain a glycerol and 2 fatty acids and a phosphate group. Hence this one is wrong. Number 3, they're used as thermal insulation. That is correct, because they're used as, for example, in seals, they're used as blubber for thermal insulation in the cold. So hence for 10 is answer C. Number 11, the diagram shows 3 different bonds. Which bonds are found in proteins? Hydrogen bonds. This first one shows, because it's between a hydrogen atom and oxygen. Any bond between hydrogen and fluorine or oxygen or nitrogen is a hydrogen bond. So this is a hydrogen bond. This is amide linkage, as you can see from this functional group C11ONH. This bit over here indicates as an amide linkage, and this is a disulfide bond. So hence all of these are found in proteins, and our answer for 11 should be A. 12, which describes the quaternary structure of a collagen molecule. For a collagen molecule, the quaternary structure can be visualized from this diagram over here. In this collagen structure, the amino acids are first arranged, and then the collagen molecule consists of 3 polypeptides, orange, green, and blue. 3 polypeptides which are forming a helix structure. These are held together by hydrogen bonds, and hence then they form the collagen fiber. So first you got your collagen molecule consisting of 1, 2, 3 polypeptides held in this helix form, which then form the collagen fiber. These are held by hydrogen bonds, by the way. So the answer for 12 should be D. The diagram shows the structure of an enzyme amylase. Which level of protein structure results in folding of amylase into a globular shape? This globular word over here is giving you a hint that this is talking about tertiary structures, because enzymes are also globular, and all of enzymes are tertiary structures. So hence for 13 our answer should be C, tertiary. 14, which statements explains why large volumes of water maintains a constant temperature when the air temperature increases. So this is a textbook definition of example of basically the specific heat capacity of water. The high specific heat capacity of water helps in large volumes of water being maintained at a constant temperature. So answer for 14 should be B. By specific heat capacity, I'll just run through it. It is basically the amount of heat required to raise the temperature of a unit mass of water by 1 degree Celsius. So that is what maintains a constant temperature of large volumes of water, which is basically very important for life as it is. So yeah, for 15 we've got which statements or the mode of action of an enzyme are correct. Some enzymes have region of the surface to which substrate has a complementary shape. Yep, that's known as the active site. Some enzymes in the substrate can change shape slightly as the substrate enters the active site. This is also correct. This is part of the induced fit hypothesis. Enzyme permanently changes its shape when they react with the substrate to form an enzyme substrate complex. That is wrong. Yeah, that's just completely wrong. That doesn't happen. Even after the enzyme substrate complex is formed, the product is formed and the enzyme returns to its shape, and the product is released from its active sites. So for 15 we got B. Okay, number 16. The graph shows the effect of an increasing substrate concentration on the rate of reaction of two enzymes that catalyze reactions with the same substrate. So which statement is correct? So for this question, we basically have to find whether enzyme Q has a higher or a lower Km value, and whether it has a higher affinity for the substrate or higher or lower affinity for the substrate than enzyme P. So first, let's just think about Km. Km is basically the substrate concentration somewhere around here at which the enzyme is working at half of its Vmax. And Km is always inversely proportional to the affinity of enzyme for its substrate. So if we talk about enzyme Q, this reaches at its Vmax value around here somewhere at a very low substrate concentration, extremely low substrate concentration, which shows that it has a very high affinity for the enzyme, for its substrate, which means it has a low Km. So all of these match with option C. Enzyme Q has a lower Km as it has a higher affinity for the substrate than enzyme P. This is because enzyme P requires much more of the substrate to reach its Vmax, whereas enzyme Q requires way less of that same substrate to reach the Vmax. As you can see, at this point, this substrate concentration, the enzyme has reaches Vmax, while enzyme P requires a whole lot more substrate concentration to reach that its Vmax. Hence, for 16, our answer should be C. For 17, the cell surface membrane structure is described as a fluid mosaic. What correctly describes the mosaic part of the cell surface membrane? The different patterns are obtained by moving phospholipid molecules. That is incorrect. The random distribution of cholesterol molecules, incorrect. The regular pattern produced by phospholipid heads and membrane proteins, incorrect. Answer is D, which is saying the scattering of the different proteins within the phospholipid bilayer. That is what describes the fluid mosaic model, basically. And we can also remember this like visualizing the proteins as icebergs in the sea. The proteins are icebergs while the phospholipid bilayer is the sea. So that's also a way of visualizing this. For 18, we've got a student immerse a plant tissue in solution S. For the next 30 minutes, the student uses a microscope to observe the cells in the plant tissue. During this time, the cytoplasm cells shrunk and no longer pushed up against the cell. This is describing plasmolysis. In many cells, a space appeared between the cytoplasm and the cell wall. Which statements are correct conclusions about these observations? The water potential in solution S was more negative than the water potential in the cytoplasm of the cells at the start of the experiment. Let us visualize this real quick. So we've got our plant cell over here, and this is solution S. The statement is stating the water potential in solution S was more negative than the water potential in cytoplasm of the cells. More negative means it has a higher solute concentration, which means it has a lower water potential. It has a lower water potential. This means this plant cell has a higher water potential. If this has a higher water potential, then water will move out from a higher to lower out of the plant cell and into the solution S, which will basically lead to its plasmolysis. So this option 1 is correct. 2 states without a cell wall to provide support, it is more likely that many of the cells would have burst. This is wrong because this only happens when water moves into the cells rather than water moving out, which basically describes turgor pressure. Number 3 states the clear space between the cell wall and cytoplasm, many of the cells was filled with air. This is wrong. It is filled with solution S, not with the air. Because of the permeability of the cell, it is filled with solution S. Hence, our answer for 18 should be B, 1 only. 19. A sample of 500 cells dividing by mitosis was examined to identify the stage of mitosis for each cell. The table shows the results. Which row is correct? The number of cells with chromosomes at the equator of the cell, this basically explains metaphase. So we take 45% of 500, which gives us 225. Number of cells with separated chromatids, this is talking about anaphase bit, so this is 24% of 500, which gives us finally 120. Hence our answer should be A. 20. The diagram shows a chromosome. What are structures P and Q? P is of course centromere, Q is the telomere. It's pretty straightforward. So that's P. 21. Which processes are used with stem cells during tissue repair? Cytokinesis. Of course, cytokinesis will be used during tissue repair because during tissue repair, many of the cells would be produced. The daughter cells would be produced, hence cytokinesis is required for that. DNA replication, of course, is needed, and of course, transcription is also needed. Both these transcription and DNA replications are linked to each other, hence, in the stem cells that are produced, the daughter cells, they will of course have their own chromosomes, and that is only possible via DNA replication and transcription. Hence, for 21, our answer should be A. 22. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. Which cells transcribe a high concentration of telomerase? Neutrophils, that is wrong. They do not transcribe a high concentration of telomerase because neutrophils are short-lived and they fight infections quickly. And for example, like macrophages, after they fight the pathogens, they die. So hence, they do not require telomerase that of high concentration at least. Mature red blood cells, they don't have any chromosomes, they don't have any organelles, hence they don't really need any telomerase. Activated memory T lymphocytes, this is the only one that's correct because they remain in the body for a very long time and they divide and increase in number when the same antigen appears again. Hence, this is basically the immune response. Hence, a high concentration of telomerase will be needed because they're going to remain in the body for a while, and when the same antigen is going to appear, they're going to divide and reproduce and increase in number. So in doing so, in order to maintain the same chromosomes, they need high concentration of telomerase. So for 22, the answer should be D. 23. Which statements are correct? All polypeptides are coded for by genes. Of course, correct. All genes are made of a sequence of nucleotides. Correct. All gene mutations result in a change to the nucleotide sequence. That is also correct. All gene mutations will result in an altered polypeptide. That is wrong, because for example, if you look at silent mutations, in these mutations, the nucleotide base sequence is altered, but it still codes for the same protein. So hence, this does not result in an altered polypeptide. So for 23, our answer should be B. 24. A double-stranded DNA molecule was analyzed and 29% of its nucleotide bases were found to be adenine. Which percentage of its nucleotide bases will be cytosine? So if 29% is adenine, then 29% will be thymine, and the sum of 29 and 29 will give us 58%. 100 minus 58 is 42%. So 42% basically consists of cytosine and guanine. So if we have 42, we will get 21%. So we've got 21% each of cytosine and guanine. So just a little recap. 29% adenine means 29% thymine, because adenine and thymine are complementary. Sum of 29 and 29, 58%. 100 minus 58% will give us 42%. That means 42% of that 100% is cytosine and guanine, because they're complementary to each other. Half that will get 21%. So 21% for cytosine and 21% for guanine each. And that'll be A. In some cells, non-coding sequences of RNA are removed after transcription. Which row correctly states the name of these non-coding sequences of RNA and the type of cell in which the non-coding sequence is removed? Yeah, the non-coding sequences are called introns, and these are removed in eukaryotes. Why are they removed in eukaryotes? Because they're removed during post-transcriptional modification, which is done before translation. So hence, this will be our answer, C. 26. Water potentials were measured inside a leaf cell, in the air spaces of the leaf cell, and in the atmosphere. Which value of water potential would allow water to move from the leaf cell to the atmosphere? So if we look into this question, the water needs to move from a higher to a lower water potential. And the higher the negative number means the more higher the solute concentration. The more higher the solute concentration means the lesser the water potential. So if we look at option A over here, we go -1. -1 means that it has high water potential. Over here in number 7, we've got -7, which means that it has more water potential. So it has lesser water potential than -1 over here, but it has more water potential than -100 over here, as -100 has a very high solute concentration. So water will move from over here, high water potential, to here, which has a bit low water potential, and over here to -100, which has the least water potential, as this has a highest solute concentration. Hence our answer for 26 should be A. I hope this made sense. If not, you can let me know. 27. 3 samples of liquids were taken from different locations in the plant and tested to measure concentration of 4 solutes. Results are shown, which samples have been taken from phloem sieve tube elements. So only thing we need to know about this question is that phloem sieve tube elements are used in translocation, and their main substance that it contains is sucrose. So the ones that contains a high concentration of sucrose, those are your answers. Since number 2 contains the least amount of sucrose, so we can just negate that option. So for 27, our answer should be C. 28. A heated band was wrapped around the stem of a plant at a height of 1 meter above the ground and left for 10 minutes. How will heating a small section of the stem to 60 degrees Celsius affect the transport of phloem sap and xylem sap between the roots and the leaves? Okay, so we should know that transport of phloem sap consists of living sieve tube elements and companion cells. They carry out this transport via active transport and of course secondary active transport, hence using ATP as a source of energy. And of course, the phloem translocation can happen in both directions, but the most important part of this question, of this analysis, is that they contain alive cells, and at this high temperature of 60 degrees Celsius, these alive cells can die. So the transport of phloem sap will be affected, and it will not happen. So it will stop. However, in xylem sap, transport of xylem sap consists of dead cells. Xylem vessel elements are dead, and causes the transport of xylem sap. And as a side note, the transport of xylem sap is caused by a transpirational pull, happens in only one direction, which is a passive process, does not require energy. And their most important point, contains dead cells. So at 60 degrees Celsius, they will not be affected by this high temperature. Hence, this will continue to occur. So for 28, our answer should be C. Moving on, 29. The diagram shows the structure of an amphibian heart and the movement of blood through it. So this is our amphibian heart. This blood is coming from the lung, going to the body, going to the lung, coming from the body. So the statements suggest which statements about differences between the amphibian heart and the human heart are correct. So the hearts have a different number of ventricles. That is correct. The human heart consists of 2 ventricles, however the amphibian heart only consists of this large one. The hearts have a different number of atrioventricular valves. This is wrong. This contains 2 atrioventricular valves. The human heart also contains of 2 atrioventricular valves, the bicuspid and tricuspid. A septum is absent in the amphibian heart and septum is present in the human heart. This is correct. The septum should be over here. However, it's absent in the amphibian heart, but this septum is present in the human heart. So for 29, our answer should be C, 1 and 3 only. A mutation in one of the genes that codes for hemoglobin changes the affinity of hemoglobin for oxygen. Okay, this sentence over here is really pretty important. This mutation lowers the partial pressure of oxygen at which hemoglobin is 50% saturated with oxygen. This means that even at a lower partial pressure of oxygen, the hemoglobin is 50% saturated with oxygen. This basically implies that the effect of the mutation on the affinity of the hemoglobin for oxygen has caused it to be higher. So the affinity is higher, hence even at a lower partial pressure, it's getting 50% saturated, which row shows the effect of the mutation, the affinity, and the position of oxygen dissociation curve. And if you look at this image over here, the oxygen dissociation curve, if the oxygen is locked in, it's going to shift to the left. However, if it is released, then it's going to shift to the right. So hence our since it's getting locked in this example, it's going to shift to the left. So for 30, our answer should be A. 31. Which processes are responsible for the Bohr shift? What is Bohr shift? Bohr shift is basically hemoglobin releasing oxygen in the presence of carbon dioxide. I'm going to come to number 1 in a bit. Let's do 2 and 3 quickly. Carbon dioxide reacts with water to form carbonic acid. Yes, this is responsible for Bohr shift. Hemoglobinic acid is formed with dissociation of carbonic acid. This is also necessary, and these both are basically linked with each other. When carbon dioxide reacts with water from carbonic acid, via carbonic anhydrase and enzyme, and this basically causes it to be dissociated in H plus ions and bicarbonate ions. H plus ions then bind to the hemoglobin to form hemoglobinic acid, which then causes the oxygen to be dissociated. So yeah, so 2 and 3 are required, but if you look at 1, the carbon dioxide reacts with hemoglobin to form carbaminohemoglobin. This is not responsible for Bohr shift because the carbon dioxide is only binding with the terminal amino groups and not with the heme iron. On the contrary, it basically stabilizes the deoxyhemoglobin. Hence, number 1 has nothing to do with Bohr shift. 2 and 3 do. So our answer should be C. Whoops. 32. A student viewed a drop of human blood with a microscope and described a cell as having a very large diameter and a U-shaped nucleus that occupies half the volume of the cell. Which type of cell is student viewing? Is pretty straightforward. If you look over here, I brought in the image. So this is basically a monocyte, U-shaped nucleus, or like a horseshoe nucleus. And yeah, very large diameter, this is occupying basically half of the volume of the cell pretty much. So it is monocyte. 33. How many times must an oxygen molecule pass through a cell surface membrane to get from the air in the alveolus to the hemoglobin in the red blood cell? Okay, assume there are no pores between the cells and the oxygen molecule must pass through. So, the oxygen first has to move through 2 membranes of the squamous epithelium of alveolus. Going out of the alveolus, 2 membranes of squamous epithelium of the capillary going into the capillary, and 1 membrane of the RBC finally getting into the RBC and binding to the hemoglobin. So 2 for the squamous epithelium of alveolus, 2 for the squamous epithelium of the capillary, and 1 of the RBC, which will give us 5. Hence our answer for 33 should be D. 34. Asthma can affect children in lungs. The air passages become narrower, less severely restricting the flow of air into the lungs. Salbutamol is a drug that widens the air passage to relieve the symptoms of the asthma. How does salbutamol relieve the symptoms of asthma? It decreases mucus secretions in terminal bronchioles. That is wrong. We need these mucus secretions. It decreases surface area of the alveoli. That is also wrong. A high surface area is vital for ventilation. It causes recoil of elastic tissue in bronchioles. That's also wrong. And it relaxes the smooth muscles of bronchi and bronchioles. This one is correct. Relaxation will cause it to become the air passage become more wider, and it will reduce the restriction of the airflow into the lungs. Hence our answer for 34 should be D. 35. The photomicrograph shows a transverse section of the trachea, which letter represents a tissue that can contract and relax to adjust the diameter of the airways. Contract and relax is basically implying muscles. So if we look at our options, A is basically ciliated epithelium. You can even see the little cilia over here. B and C are basically connective tissues, loose connective tissues, so they aren't really responsible. And these little circular structures over here are mucus glands, by the way. So D is the correct option because this part over here is the smooth muscles, which are responsible for the contraction and the relaxation. So 35 D. 36. Which statements about an infectious disease may be correct? It can be caused by productis, that is correct. It can be transmitted by insect vector, that is correct. An example of both of these is basically malaria. It can be transmitted from mother to child, that is also correct. An example of this is HIV. So for 36 we've got A. 37. Which statement explains why cholera has not been eradicated by vaccination? There is limited availability of an affordable vaccine, that is correct. Protection takes several weeks to develop after vaccination, correct. The underreporting cholera in the community, correct. and the immunity cholera decrease after 2 years. That is also correct. All of these options are basically examples, reasons why cholera has not been eradicated by vaccination as of yet. So should be A. Even if we don't know this point, immunity to cholera decreases after 2 years, we should hypothesize that why is vaccination not helping against cholera. So if the immunity cholera decreases after 2 years, this is basically explaining the question itself. This is occurring, which is basically leading to vaccination being of no use. So yeah, even if we don't know this, we can decipher it. So A should be our answer. 38. Antibiotic resistance is a serious threat to global health. Which statement describes a statement taken to reduce the impact of antibiotic resistance? Antibiotics prescribed by doctors should be shared with family members to protect them. This is wrong. Antibiotics should only be used to treat infectious diseases caused by viruses. No, it's used against bacteria. Farmers should give antibiotics to healthy animals to prevent infections occurring. Nope. We only give them after the infection occurs. Vaccination programs should be used to reduce the spread of bacterial diseases. This is correct. So D. 39. Tetanus is an infectious disease caused by a type of bacterium. Bacterium produces a protein that is toxin which causes illness. Produce a vaccine for tetanus which contains a harmless form of toxin called toxoid. Toxoid is remixing toxin with the chemical formaldehyde. This chemical binds the toxin, making it harmless, which statements why the tetanus vaccine is correct. After vaccination, the toxoid in the vaccine will be ingested by neutrophils and displayed in the cell surface membranes. Yep, that's basically a textbook example of a vaccination, of a vaccine basically. So answer should be A. So after the neutrophils carry out this antigen presentation, basically this antigen presentation of the antigens, these are they basically received by the T helper cells. The T helper cells will recognize these antigens as non-self and will cause an immune response against this toxoid, which is displayed. Yeah, so this will cause an immune response, activating all the macrophages, the B lymphocytes, the plasma cells, and most importantly, memory B cells. If these memory B cells will basically remain in the body for a long time, and if the same antigen comes into the body, this time in its actual form, not in the form of vaccination, this will basically the B lymphocytes, memory B cells will basically recognize this antigen as non-self and will initiate the immune response very quickly, releasing a high amount of antibodies which will basically protect your body. So last question, number 40. What is an effect of the immune system of reduced number of T helper cells? An example of this is HIV, by the way. And an effect on the immune system of a reduced number of T helper cells is basically a decrease in the activation of B lymphocytes, which I just explained right now. If there's less activation of B lymphocytes, less plasma cells, less antibodies released, most importantly, less memory B cells. And there's less memory B cells, this will basically prevent long-term protection towards your body against all these antigens, non-self antigens. So immune response will, the immune system will be compromised with a less number of T helper cells. Hence, very important. Which an example of HIV, and basically this reduced number of T helper cells in HIV, for example, causes many opportunistic diseases to occur in the body, like Kaposi sarcoma and TB, pneumonia, all that. So yeah, so answer for 40 should be B. This concludes our paper. Hopefully you all enjoyed and understood it well. If you didn't, you can always let me know. So yeah, let me know if this was a good video or not. Yeah, I'm always trying to improve. So yeah, take care. Goodbye.